Welcome, BrookingsBiology students. This is Mrs. Rydell, and in this slideshow, we're going to look a little closer at some of the changes that can happen to DNA. We talked about mutations a little in our human disorders chapter. We're going to look a little bit about what's actually going on with the DNA when mutations happen. So if you remember, mutations are changes in the genetic material, change in the code. And mutations can happen for lots of reasons. They can happen when cells make mistakes when they're copying the DNA during mitosis or meiosis. They can be caused by radiation or other chemicals in the environment. We talked about different carcinogens in our mitosis chapter, and in our chapter when we talked about some genetic mutations in humans. Environmental factors can change the DNA code in a cell. We talked about viruses, we talked about radiation, we talked about ultraviolet light, sunburn can lead to skin cancers. We talked about some different things that can cause changes in our code. We also talked about this, the kinds of mutations. If I'm causing a mutation that changes just one gene, the gene for hemoglobin, the gene for blood clotting proteins, the gene for making an enzyme to break down phenylalanine. If it's a single a mutation in a single gene, we call it a gene mutation. Sometimes mutations cause changes, bigger changes to whole chromosomes, maybe pieces, a whole arm missing off a chromosome, or a missing chromosome, or extra chromosomes. We call those chromosomal mutations if it changes more than one gene. So let's look at some of the changes that can happen a little more closely. If it's a mutation that just involves one or just a few nucleotides in one little place on the DNA, a single point, it's called a point mutation. One point, one little spot is changed. And we have a couple different kinds of point mutations. Substitutions, deletions, or insertions. Putting in an extra, removing one, or changing one that's there to something else. We're going to watch a little video about some point mutations that can happen. We're going to watch a little movie about some of the changes that can happen because of point mutations.
The human genome contains billions of pairs of nucleotides. A base pair substitution occurs when one of these pairs is accidentally replaced by another. For example, during DNA replication, this adenine-thymine pair could be accidentally replaced with a guanine-cytosine pair. Some base pair substitutions are harmless. Although these substitutions change the DNA sequence, they don't alter the resulting amino acid sequence. For example, consider the DNA sequence CCG. When transcribed and translated, this sequence specifies the amino acid glycine. What would happen if the last nucleotide of this DNA sequence were accidentally changed from G to C? When transcribed, the new DNA sequence would produce a different messenger RNA. However, because the new RNA sequence also codes for glycine, there would be no change in the resulting protein. Other base pair substitutions can be deadly. A single base pair substitution in the normal gene for hemoglobin can produce the gene for sickle cell hemoglobin. Transcription of the two genes produces two different messenger RNA sequences. When the two RNA sequences are translated, they produce proteins that differ by one amino acid. Under certain conditions, the mutant protein produced by the sickle cell gene causes red blood cells to change shape. The result is the painful and life-threatening symptoms associated with the disease sickle cell anemia.
So a substitution now is changing one base, replacing it with another one. So if you look at the code, here's my original strand, and if I cause a change in just one of those bases, I switch the letters. Instead of a G in this spot, I change it to a T, and it can happen during a copying error when the cell is copying its DNA, putting in the wrong base in that spot. And this would be what we call a substitution. If you think about where we use that other word, substituting something, we're replacing the one that's there with something different, like when you have a substitute teacher. And substitutions, even small ones, can cause big changes in the gene code and then in the protein that is produced. We talked about sickle cell anemia, and that is a substitution. You're changing one letter, but it changes the amino acid from glutamine to valine, and it changes the sequence of the protein. And it causes a change in the hemoglobin protein, the way it acts and how it carries oxygen. And we talked about sickle cell and how the blood cells that contain that hemoglobin change shape and can cause kids to become very seriously ill. We talked about that in our human disorders chapter, and it's caused by a substitution, changing one letter for another. Deletions are pieces of code where a section of the code, either one letter or pieces on a whole chromosome, are lost. About deletion, when we use that, talking about computer program, if you're typing a paper for English and you highlight and delete something, hit that delete button, you're removing that little section of the writing code. And that's what happens to the gene code. If we take this, here's my original piece of DNA, and if these stand for the different genes, remember, lined up on a chromosome, if there's a little section that is removed, deleted, that is going to change the gene code. The information that's coded in that section is going to be missing in my cell. We talked about muscular dystrophy. It is an X-linked recessive disorder caused by a change in the muscle protein coding gene, and it's a deletion. There's a little piece of the code that's missing in the gene that codes and tells these kids how to make muscle proteins, and it causes a serious genetic problem. We talked about this in our genetic disorder chapter. This is a genetic disorder that's caused by a deletion. Adding extra pieces, extra copies or extra letters into the code can also cause some problems. That's called an insertion. We're putting in extra pieces, and if the insertion is multiple copies of the same code, we call it a duplication. We're duplicating more than one copy. If we're adding in extra code, that's called an insertion. When you insert something, it means you're adding it. Gene mutations, if it's a substitution, generally we're going to just change one amino acid because we're just changing one letter for another. Remember, we read our code in little groups of 3. So if I change one letter, it's going to change that one amino acid that little 3-letter group is coding for. So substitutions may change one amino acid. If we put in a deletion or an insertion, that can have a way bigger effect because adding extra letters to the code or removing letters from the code causes a change in the little groups of 3 that we're reading. So adding in an extra letter causes what we call a frame shift mutation. Frame shift mutations cause more serious problems because they affect more of the code than a simple substitution. If this is my, we're going to kind of make this into a sentence so we can kind of see what's going on rather than just looking at A's and T's and G's and C's. If this is my piece of DNA now, and I'm going to read this in little groups of 3s to make a message for the cell to read and turn into a protein. I would read this as the fat cat ate the rat. That makes sense, right? There's information there that I can use. If I add in an extra copy of part of that code, and I read now, the fat cat ate the rat. That would be what we call a duplication. We have extra pieces. I can still kind of tell what is going on in this little story, right? What if this is my code now and I remove one of the letters? What if I remove the C here? It gets cut out and by mistake when the DNA is copied. And now if I read this in little groups of 3, this little code is missing. It's going to move everything over one space. And now the little groups of 3 are going to be grouped differently. The fat, oh, now my C is missing. This becomes my new group of 3, and this becomes, and now if I read that information, the fat tetherat, oh, I've lost the code. I've lost the information. I'm getting a different, totally different message. And so if I cause what we call a frame shift mutations change every amino acid in the sequence of the protein that follows where the shift happened. And frame shifts can change a protein so much that it is unable to function. If you change not just one amino acid in the sequence, but totally change the amino acids that come in the sequence, that is going to produce a totally different protein, maybe one that is not going to be able to do its job. If you look at where the mutations happen, the location of the shift is important, right? If the shift is near the beginning of the gene sequence, I take out the T, for example, everything after that mutation is going to be changed in my message, right? I change a whole lot of that information. If the mutation happens near the end of the gene, the fat cat ate the, what if I lose this part of the code? It's toward the end now, and everything up to that point is still going to be read correctly. The part that's going to change is just the little bit that's after the frame shift. And so mutations, frame shift mutations that happen at the beginning of a gene are going to damage more of the code than ones that happen at the end of the gene. We talked about chromosomal mutations when we did our human disorders chapter. Remember, mutations, if I'm changing the number of chromosomes, adding chromosomes, missing chromosomes, if I change the structure of whole chromosomes, maybe deleting part of an arm off of a chromosome. And so we're going to look at a couple different kinds of chromosomal mutations. Deletions, duplications, inversions, translocations. Let's watch a little video about how this might happen in a cell.
Crossing over is the exchange of fragments between homologous chromosomes. During prophase 1 of meiosis, homologous chromosomes lie close together. This is when crossing over occurs. However, if the arms of the chromosomes are not lined up evenly, breaks may occur in different places on the two chromosomes. This produces two different sized pieces. The longer piece contains an extra gene that the shorter piece lacks. The different sized pieces may be exchanged. When the short piece of chromosome is added to the shortest broken arm, a gene is left out. Here, the gene, represented by the blue band, is missing. This type of mistake is called a deletion. The longer piece contains genes that were already present on the other chromosome's arm. When the long piece is attached, the chromosome gains a second copy. This type of mistake is called a duplication. When the pairs segregate and the chromatids separate in meiosis, 4 chromosomes are produced. 2 of them have altered structure as a result of unequal crossing over.
The other kind of mutations we talked about on our list were translocations and inversions. So let's watch a little movie about that.
The number and arrangement of genes on a chromosome can be changed by translocations or inversions. non-homologous chromosomes line up next to one another. A piece of one chromosome may break off and become attached to the other nearby chromosome. As a result, one chromosome gains genes and the other loses them. This kind of mistake is called a translocation. The genes on a single chromosome can also be rearranged. A fragment of the chromosome may break off, turn completely around, then become reattached. This rearrangement is called an inversion.
Let's get this down in our notes. We talked about deletions, a base pair deletion as a gene mutation, but if we delete a bigger section, multiple genes being removed instead of just one letter in the code being lost, we could have a chromosomal deletion, and that is going to cause a piece of the code containing those genes to be lost from the gene code, from the DNA, and it's going to result in a cell that's missing important information. A duplication happens when a cell gets too many copies of the same gene. So this CD gene, we're making multiple copies of that, and it happens during mistakes with crossing over. We saw in the little video how one cell can get two copies of the same section of gene if the DNA crosses over unequally, and we end up with a chromosome that has multiple copies of a section of code. We talked about Huntington's disorder when we did our human genetics chapter, that brain disorder. We talked about the song, This Land Is Your Land, and we talked about Woody Guthrie and writing the song and how he ended up with developing the symptoms of Huntington's. And it's caused by additional repeats, extra CAG, 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 CAG repeats in the gene code. This would be called a duplication. Inversion, when one of the DNA breaks and the code flips around backwards and reattaches in that position, leads to something called an inversion. And inversions can also lead to other kinds of mutations. Once the inversion is in place, it can lead to duplications and unequal crossing over and missing pieces in our DNA. We're going to watch a little video about that.
An inversion is a reversal in the order of a segment of a chromosome. Inversions do not alter gene expression unless the breakpoints on the ends of the inversion occur within a gene. When a segment of a chromosome is inverted, it can pair in meiosis only by forming an internal loop. Although crossing over occurs in the 4-strand stage during meiosis, only 2 strands are shown for clarity. Crossing over that occurs within the inverted segment during meiosis can result in some genes being lost on one chromosome, while others are duplicated on another chromosome.
Inversion can lead to other kinds of mutations, deletions, and duplications. The other type of mutation that you saw in the video was exchange of DNA between chromosomes that are non-homologous partners. Remember, in meiosis, homologous partners cross over and trade genes, but crossing over is not supposed to happen between chromosomes that aren't homologous partners. If a piece of DNA from this chromosome breaks off and attaches to a different non-homologous chromosome, you can end up with what we call a translocation. It's moved its location to a new place in the DNA. Those genes now are going to be carried on a different chromosome, and that can cause some problems with missing an extra code as well. Most mutations are what we call neutral mutations. Little or no effect on the gene and how it functions. Remember, we talked about that wobble built into the message code, and how even sometimes just changing that one last letter still puts in the same amino acid into the sequence. Mutations that cause proteins to be made in a different way are usually harmful. If you make the protein so it can't do its job, so it doesn't function the way it's supposed to, that usually leads to some kind of a genetic disorder. And harmful mutations are often associated with those genetic disorders or with cancer. We talked about mutations that can lead to cancer. Some mutations, though, are beneficial to organisms, and we talked about this also in our human disorder chapter. Mutations that cause a change in the organism that can help it to survive and reproduce in its environment. Example would be having a lighter color, a white fur, if you lived in a cold, snowy place. That kind of mutation could allow an organism to stay alive, blend in better, has camo, maybe not be eaten as easily, or if it's a predator, could sneak up on prey and get more food easily. If you have a mutation that increases the chance of an organism's survival and ability to reproduce and pass that gene on, that is going to be what we call a beneficial mutation, and it provides variety in the population. Think about all the people in this room, different skin color, different hair, different height, different eye color. Everybody's different, and that variety in the population is the basis for natural selection to act on. Remember in our first chapter, our populations change over time, that idea of evolution. We're going to talk more about that second semester. But some mutations can actually be beneficial mutations. Another kind of mutation can happen when a cell copies its DNA but doesn't go on to divide. Those extra chromosomes, extra sets of chromosomes, stay in the cell, and it can lead to a condition called polyploidy. Polyploidy organisms have extra sets of chromosomes. Remember we talked about a sperm or egg having a 1N number of chromosomes, one copy of each of the chromosomes. 2N cells, body cells, somatic cells, have 2 copies of every chromosome. We call those 2N organisms. In humans, having polyploidy, multiple sets of chromosomes, is lethal. But in plants, some kinds of plants, having multiple sets of chromosomes, 3N, 4N organisms, even like 8N, 6N, often in plants, being polyploidy is a benefit. Those plants are larger and stronger than diploid plants. They produce more fruit. Some of the foods that we eat, think strawberries, watermelon, those are polyploidy plants, and they have extra sets of chromosomes. That's the end of our little slideshow about mutations, and stay tuned for one last section in your chapter about gene regulation.